Screen sharing. Got that. All right. And can you check on your and make sure it's broadcasting? I mean, I'm sure you can't hear voice. My headset on. Ooh, it's more complicated than it needs to be. How are you? Good. How'd your field work go? Did you collect some nice flowers, or good? They were just a little late blooming, eh? Okay. But you got it done, and awesome. So you guys, please help yourself to some fresh fruit that I brought in the back. I'm glad Stephen went ahead and did that. And even if you don't want it right now, you need to take some for later because I can't eat all that. Does it work? Okay. Awesome. Look at this quiz we could take for fun. We could label places in the Mediterranean region. We could answer some questions about the history of the Roman Empire. This is like, I'm surprised how much I remember. This was left behind. I'm excited. Yeah, we typically do, which is nice for our department that we're small enough that we can do these kinds of seminars during the regular time, because it certainly improves the number of people that are able to attend because people have already set aside time. But of course, in a large department, like, so there's absolutely no way that would work. You wouldn't do anything else but these kinds of seminars. Huh? No, he's back. He's back. He is back. So how are you? So here's a question for you. I found us made this quiz that was left behind. It's really important. This is all about the Roman Empire and things. Look it up. I'm so shocked at how much of this I remember. Wow. Well, I was commissioned to make you remember, so that might have helped. That's where it's like, these are familiar to me now because I was last semester. Yeah. See, this is, what are you bored during the seminar? You can entertain yourself, take this quiz. Oh, thank you very much, sir. I really appreciate that. Oh, and so please help yourself. I brought some fresh fruit in the back, so even if you don't want it now, pocket some and take it for later, please, because it won't. I can't possibly eat all that. Offer it to the horse. She can have an apple. Yeah. She would love it. Not the bananas. Actually, she didn't eat the bananas, but I'm not sure how much I agree with her. I think our dog got into some onions yesterday. Oh my goodness. So not much. I mean, I'm thinking just like little tiny pieces that probably hit the floor, but he has been real sick. So I'm sure I'm hoping it will pass. So do you have it right now? Can I just quickly do a sound? Check on that. Oh. So just like I've got to stand back here the entire time and use this headset to broadcast. So just so you don't think I'm like hiding back here behind the. Oh yeah, that's exactly what it's going to look like. Oh, it's on the eat. You don't want it now for later. I mean, I can't eat all that, so please put it in your bag, take it home.
Because by the end of the seminar, I won't be fresh anymore.
Well, if the ethylene content in here becomes as high as it was in the
Is that growth in the cooler? Watch out for ethylene. Has that mystery been solved?
I don't know. I did contact Armand about it because I thought that maybe it was 1404, and we actually have a refrigerator in 1404, so I wasn't.
Actually, I shouldn't ask that too loud. Embarrassing.
Oh.
Guilty parties.
You win bonus life or That's an important part.
If I could have a volunteer timekeeper so that members, I and other members of the committee can focus on the presentation. Sorry, how long is it? Yeah, at 20 past, I'm gonna give her, sorry, at 10 past, give her some sort of sentence. Okay. And then at 20%.
That would be really well. Keep in mind, I'm going to be wearing a headset, so you have to. Cawing is not going to.
Yeah. What you need is a remote that sounds an electric shock.
You might be surprised at how effective that is. Might be surprised.
How many remote people are there?
I don't know yet.
The whole internet.
Well, and technically the whole internet could join, which is why I'm not allowing for remote questions, because I've heard that these kinds of seminars are sometimes trolled by people who ask really inappropriate questions.
Well, we'll keep the inappropriate questions to this one.
Yes. It's actually two committee members are somewhere else, and I think they're both joining for this, and only one will join us for Skype later on. And then some friends and colleagues. And, so those of you who have come and are realizing now that you could go back to your office and get the same effect, by all means.
So Amy has kindly provided some fruit. So you're welcome to have yourself.
Yeah, and if you don't want it now, you definitely want it later, I assure you.
Just be careful where you bring the ethylene. We start with a moment of silence. Okay.
We need a moment of silence still at this
So,
sink.
I think we're ready to go. I don't know where everyone is, but it certainly is a busy time for the university. I know that when we have distinguished visitors coming, that often the introduction spans well into 20 minutes or so. This is one of those rare cases where if I did give a full introduction, it probably would take a lot of AJ's time, and she has a lot to tell you. So I will abbreviate it. But needless to say, she has accomplished quite a bit during her stay here so far with peer-reviewed publications, awards for those publications, grants both internal and external, travel internships, fairly high prestigious ones. And well, I could go on. And she has a postdoc lined up, and a federally sponsored trip to China lined up as well. So, so truly amazing set of accomplishments for a graduate student. She got her master's at NC State in 2007, and I'll just quickly go through this because, as I said, it could take a long time to list all her accomplishments. She joined OSU in 2010 under Linda Watson's direction, but she developed an interest in bioinformatics and biodiversity and multivariate quantitative methods for analyzing biodiversity data. So she joined my lab several years ago. Yes, I have lists of awards and accomplishments here that I'm going to not present. But I will say that she does have a background background in phylogeny, an interest in paleoecology, interest in phylogeny and systematics, and so she is broadening it into biodiversity. Because her newfound area is largely interdisciplinary, she spent quite a bit of time working on methodology. So a lot of what her presentation will be about today will be basic methodology in the field of linking paleo to modern biodiversity. Even though it is largely methodological, you will see a fair bit of science shining through. And I'll let AJ make the case for why this is an interesting discipline, and also why it's a troubling and problematic. It's important by itself in major scientific events. So, or message, I should say.
So thank you for that, Dr. Palmer. And I'm just letting you guys know in advance, I'm standing back here with this headset on. We do have remote viewers, and so I need to be here so I can discuss with them. I don't have a Bluetooth headset. I will also be using my mouse as a pointer today, as opposed to a laser pointer, so just kind of be on the lookout for that as I zoom around my slides. Okay, so I've used some of your pictures here without permission, and I truly apologize for that. If you're offended, I hope you're not. What I wanted to begin was by saying that these have been some of the most eventful and important 5 years of my life. And one of the most exciting things about being here at Oklahoma State University has been the opportunity to interact with some really amazing biologists, people both in this department, in the botany department, elsewhere on campus, and then outside of the OSU community through grants and fellowships. So these people are people who have agreed to generously serve on my committee, have taught courses that have been absolutely fundamental to my development as a researcher, people who have co-authored publications with me, and people who have mentored me in a myriad of ways. One of the other really cool things about interacting with this group of people, and others, I'm sorry, not everybody's picture could make it up here, is that our interests, even in this botany department, represent a real diversity of fields. And so, we have people who work in the areas of taxonomy and systematics, people who work in genetics, in plant development. It really spans quite a broad spectrum. And so I put up here this phylogenetic tree to remind us that as biologists, the thing that really links us together and underpins our research and the hypotheses that we develop is the fact that life on Earth evolved. This phylogenetic tree here shows that life evolved over quite a long period of time. As a biological scientist, my niche within my niche within biological sciences, I guess you could say, is in the area of biogeography, so how organisms have evolved in space. And so you guys know that I'm really fond of biogeography. In fact, you might have even been a little bit stunned there that biogeography didn't make its way into the title of my presentation. Well, don't be alarmed, because as it turns out, past and present plant distributions is really just another way of saying biogeography in kind of a wordy humanities way. So, and I say humanities way because I can say that because my background is, my undergraduate background is in humanities. Okay, so in biogeography, we're interested in how shifts in the ranges of organisms on the face of the Earth as they evolve. So they interact with abiotic factors or biotic factors to undergo things like local extinction and range expansion on very small time scales, and at very large time scales, things such as vicariance and long distance dispersal. So in a traditional study of biogeography, biogeography has been divided into sort of two subdisciplines. A discipline that looks at the small scale interactions, so local extinctions and range expansions. I don't know what's going on with my mouse. There we go. Okay. And a discipline or a subdiscipline that looks at large-scale reactors or reactions over a long period of time, such as vicariance and long-distance dispersal. I would argue that actually those distinctions or that compartmentalization is a bit arbitrary. And in fact, I've been very fortunate to have had committee members here at Oklahoma State University who have really encouraged and mentored my interest in biogeography across those timescales and let me investigate lots of interesting topics within biogeography that weren't confined to either the short timescales or the long ones. So I just want to go ahead and acknowledge and thank my committee for their mentorship and their putting up with me over the course of 5 years. So that would be my advisor, Dr. Michael Palmer, and committee members Andrew Doust, Mona Papish, who cannot be with us today, Jeanette Steets, and Dr. Jen Nguyen, who will be joining us remotely for this talk as well as for a Skype later. And so thank you to all of those committee members. They have fostered a project for me in 4 distinct parts that has allowed me to explore a variety of topics within biogeography. And so I'll briefly go over those with you, and I'll use this slide as kind of a roadmap for where I'm going today. So this dissertation, again, has had sort of 4 distinct parts, 4 questions or 4 objectives, if you will. And don't worry if some of these questions sound a little bit jargony right now, because I'll tell you what you need to know before I launch into the details, okay? So first of all, do ecological niche models accurately predict biodiversity at different geographic grain sizes? How does taxonomic rank affect the predictive power of paleoclimatic modeling? What paleoclimates did Aeschylus experience during the Eocene? And lastly, was there a latitudinal diversity gradient of genus richness of vascular plants during the Eocene and Oligocene epochs? So these first 3 questions are really concerned with using models to understand biogeographic topics, while the last question is concerned with investigating a classic topic in biogeography. So without further ado, I'll launch into this first objective and talk to you briefly about a background and then more about methodology and results. Throughout this talk, I want to acknowledge co-authors of the studies that I'm presenting here. So each of these 4 objectives is in some state of publication at this point. That is out the door and already published, under review, or hopefully ready to go out the door very soon. And so I'll just, for each of these objectives, I will just acknowledge those co-authors. And this objective has resulted in a paper that is currently under review with co-authors Mona Papish and my advisor, Mike Palmer. In order to really understand what I'm getting at here and why this is important, do ecological niche models accurately predict biodiversity at different geographic grain sizes, we probably need to define a few things. So let's start with ecological niche models. Now, some of you are quite familiar with that topic, others of you may be less so. But generally, ecological niche models can tell us something about, or can predict, the distribution of a taxa or of a taxon, or where that taxon might like to live, where it might, where habitable environment might be for it. And so the typical way that an ecological niche model functions is by taking known occurrences of a taxon. So I'm kind of struggling with the mouse here. Let's see, can I? No, okay. So taking known occurrences of a taxon, so these little green points here are actually in the state of California. I don't know how well you can see that there. And they each represent some kind of observation, maybe an herbarium specimen, a note in somebody's field notebook about where a particular taxon of interest occurs. Each of those points in geographic space has a climatic dimension, meaning that at that exact point in geographic space where that taxon was observed or collected, there is something known about the isothermality of that area or about mean annual temperature. We can collect climatic variables from those, the locations of those occurrence points. We can combine that information, that is the occurrences of the taxa, with the climatic data using a modeling algorithm, and the output is some predicted distribution of the taxon or a predicted suitable habitat for the taxon. And that's shown here, where warm colors represent areas where that taxon either does live or may really like to live, where cool colors it either does not live or probably doesn't like living. Okay, so I want to use ecological niche models to predict biodiversity. Well, biodiversity is a buzzword. You guys hear that all the time. And so therefore, I feel like I need to define it because different people mean different things by it. In this case, I simply mean species richness. And so species richness is the number of unique species that live within a well-delimited geographic area. So here, this black box, I'm showing you a well-delimited geographic area. And inside, I am showing you that 4 unique species occur there. So the ideal way to determine the number of species that live in a unique geographic area would be to go out and count them. However, you can imagine that even on the scale of a 50-meter plot, a vegetation plot, that task becomes quite difficult. Now imagine counting the number of species that live in a place like Payne County. So I want to find, or there's a lot of interest actually in finding ways to estimate that diversity, particularly for areas that are difficult to access or have not yet been surveyed. So again, I want to use ecological niche models to accurately predict biodiversity or species richness. And at different geographic grain sizes, what does that mean? Well, that's just the size of the black box. So the area in hectares or whatever. Why does geographic grain size matter? Well, a lot of work has been done on this topic. Can we use ecological niche models to predict species richness? But it's mostly been done at very small geographic scales, on the scale of a meter or two. So I want to know, can we scale up? Can we predict the richness of a place like Payne County, the top of a mountain in the Andes, or something else that might be a little bit more biologically meaningful under certain circumstances? Okay, so I've showed you how niche models function to predict the distribution of a species or the potential distribution of a species. But how can we predict biological diversity or species richness using ecological niche models? So I've already walked you through this scenario here. You take a taxon, take its occurrence, take some occurrence points for that taxon, extract the environmental data from where those the location of those occurrence points, apply a modeling algorithm, and get some model of species distribution within a geographic area of interest, represented here by this black box. So actually, I can do that for a whole lot of taxa, such as these here. I can then project that onto some geographic area of interest. And then for any delimited space within the geographic area of interest, represented by this flashy box here, I can determine the species richness. Or at least that's the hope, right? And so here, if you see the number of models that overlap within the flashy box, that's 2 models, and therefore we would have a species richness of 2. So if I want to test how well this works, are there really 2 species there? I need a couple of things, right? So I need some taxa. I need a study area. I need well-delimited spaces within that study area. So here's a study area in this black box. Here's a well-delimited space within the study area, the one that flashed. I need occurrences of the taxa, such as this, shown here. I need environmental data, and I need a modeling algorithm. So let's take a look at where all that's coming from. So in terms of taxa, I decided to study Cactaceae and Pinaceae. Now, why these taxa? Well, here you're going to see that they are widely distributed in North America, north of Mexico. And so I chose North America, north of Mexico, which I'll lovingly call North America after this. So that is my the geographic extent of my study region. And the Cactaceae and Pinaceae are well distributed within that region, widely distributed within that region, evidenced by the amount of green coverage shown here in these maps, Cactaceae on top, Pinaceae on the bottom. However, within North America, we know that their distributions are really quite different. They're obviously responding to very different, or they're obviously responding quite differently to their environment. So these represent 2 really different study systems. And that's evident based on the concentration of species of Cactaceae, which is mostly here in the American Southwest, and concentration of Pinaceae species, which is mostly here in the American West and the East. And I only have data here for the United States. Okay. So now I know that North America, north of Mexico, or again, North America, is going to be the full extent of my study region. Now I need some well-delimited spaces within North America in order to look at the predictive power of ecological niche models for species richness. And so what I did was I used published floras, or the areas delimited by published floras. Those are floras that are curated within the Floras of North America project, and that project is curated by Dr. Palmer and some of his colleagues. And it simply reports some meaningful data for floras, or records records for floras that meet some minimal information criteria, especially having well-delimited geographic space. And so in order to look at this topic, or in order to look at the ability of these niche models to predict biodiversity at different geographic, or to predict biodiversity at different geographic grain sizes in North America, the grain size was important. And so I designed a stratified sampling of these floras from the database, such that I sampled a total of 180 floras, representing 6 different geographic grain sizes. Those range from really tiny, we're talking under 10 hectares here, to pretty big, up to 10 million hectares. And then to make sure I fully covered the land area of North America, I stratified sampling according to an Arctic and Atlantic and Pacific region, again, for a total of 180 floras. Now, these floras also provide a really, so you might be thinking to yourself right now, well, why predict the number of species of Cactaceae and Pinaceae within these published floras? Because you already know it. Well, yeah, that's part of the point, right? So here we can use the floras as a test of the accuracy of the stacked niche models. Okay. So let's just think about how that's gonna work, how I'm going to get those stacked niche models and make those predictions. So for the North American Cactaceae and North American Pinaceae, I have some taxa. These are all taxa coming from those floras that the 180 floras. For each of those taxa, I obtained some occurrence data that's coming from the Global Biodiversity Information Facility, or GBIF. Some environmental data from these global sources. A modeling algorithm, in this case I use Maxent, for those of you who are familiar with the different modeling algorithms. I ended up with some predicted distributions and constructed some stacked models independently for each of these plant families across the North American region. Whoops. And then within the North American region, I looked at what was going on within the individual floras. There's 180 floras. Okay, so let's talk briefly about some results. Now, I want to call your attention here, just this is a very complicated slide, but pay attention to the graph highlighted here in yellow. So what you'll see is that on the x-axis, I'm showing you the observed number of species. On the y-axis, I'm showing you the predicted number of species. This is for Cactaceae, okay? So observed on the x, predicted on the y. And for this first graph, I'm showing you all the floras, regardless of size class, just lumped in together. So this dotted line represents a regression line. And you can see an R² value up in the corner there of the chart. And the dash, or rather the solid line, represents a line of equality. So line of equality meaning data points on that line represent perfect predictions. So that is, we have one species in a flora and we predict that there is one species there. Anything above that solid line represents an overprediction, so we're predicting more species than are actually present. Anything below that solid line represents an underprediction. So takeaway message here, R², not so high, and most of these, for most of the floras, there were over predictions of species richness. Now, if you look elsewhere in this graph, or elsewhere in this figure, what you will see is that I've actually divided up, I've done the same regression, same scatter plot, same regression for the floras of different size classes. So here, very small floras, and up in size through here to the largest floras over here. Now, two things I want to point out here. Thing one is that consistently we see that there is an overprediction of species richness for Cactaceae. And as we go up in size class, generally speaking, the correlation between predicted versus, or between observed versus predicted improves. Now, we're still way over predicting, but there's a better correlation between the preserved or observed and predicted. Not so for Pinaceae. So very similar graphs here, highlighting in the top corner the graph indicating or for all of the size classes lumped together. So you'll see two very different things about Pinaceae here. First of all, there are a lot more underpredictions. Secondly, although there is some increase in the correlation between observed versus predicted, that increases much more slowly, and R² here for the largest size class of flora is still quite small in Pinaceae. So let me back up just a second and say, up until this point, I've only been talking about numbers. I've not been talking about taxonomic identities, okay? So even if you got the numbers right, you could still get all the taxa wrong, okay, in terms of predicted or what the models predict to be present in a particular flora. So we can also think about the accuracy of these ecological models in terms of, do they correctly predict the correct taxa in the floras? So here in these graphs, I'm showing you on a logarithmic scale on the x-axis, and then Cactaceae at the top, Pinaceae at the bottom. Logarithmic scale on the x-axis representing the size in hectare of floras. And then here on the y-axis, I'm showing you a measure of similarity. And so high values are really good. 1 equals absolutely similar, and we're talking about taxonomic identity here. And 0, zero similarity whatsoever. Okay. So 2 things going on here. First of all, you will notice that for Cactaceae, these values are all very low. So even in cases where the ecological niche models did a fairly good job of predicting species richness, they're not predicting the correct taxa, right? And there's virtually no trend here across grain size. Universally doing doing poorly at all grain sizes. A little more spread here in the data in Pinaceae. Oh, and I should point out that these values here at the top, where we do have perfect scores, so perfect similarity of 1, these all represent cases of absence. So absent, thank you for laughing at that. I thought that was pretty funny too. So yeah, these all represent cases of absence, so where there are no predicted and no observed Cactaceae. So anyway, that's that. Looking down here at Pinaceae, there is more spread in the data, but certainly no trend in the in grain size. Okay, so some quick conclusions from that part of the project. The precision of model predictions of species richness did in fact improve with grain size. More so or at a faster rate in Cactaceae than Pinaceae. However, I'm going to go ahead and call it here and say the ecological niche models were not very good predictors of species richness for Cactaceae or Pinaceae in North America. And model accuracy may be taxon specific, and that's the real rub, right? Because that means that we can't have a one-size-fits-all correction for over-prediction, okay? So now I want to move on, shift gears here, and talk about this. How does taxonomic rank, a random of there, of something, affect the predictive power of paleoclimatic modeling? Really fortunate to have worked with a lab mate, Justin D. Where is Justin? Right there, on this project. And so I hope I don't butcher it up here, since, you know, he's in the room and has got to listen to me do it. Okay. All right, so I've already talked to you a little bit about, let's see, so how does taxonomic, we're going to talk about now, I have to back up, how taxonomic rank affects the predictive power of paleoclimatic modeling. And so I've already talked to you a little bit about how we can use these stack models if we really want to, and I'm not recommending it, to predict species richness. However, when we have these stacked models here, when they're overlaid like that, they don't just exist in geographic space. They also exist in environmental space. And so where all of these 4 models overlap with one another, I can extract environmental information, and I can assume that that would be a place, or that kind of environment would represent an environment in which these 4 taxa could co-occur. So now let's think a little bit about paleofloras. So why would I want to do this at all, right? I can just go out and, like, you know, measure the isothermality or the, you know, mean annual temperature of a spot. Well, in the case of paleofloras, we often don't know anything about the environment in which these taxa occurred. So this represents a way for us to measure that, or to acquire that information. So with paleofloras, we have an extra step here, okay? So we're going to take a fossil taxon, or all the fossil taxa in a flora. So here we have Aeschylus, Equisetum, and I think an Ilm. We're going to map it on to a living taxon. And then we're going to go through this process, find occurrences of the living taxon, extract environmental data, apply a modeling algorithm of some sort, get some predicted distributions, stack up the ecological niche models, and then extract some environmental data. Okay, well, that's fine, but here's the problem. So when we work on fossils, it's pretty rare that we work at the level of species. Now, that's for a couple of reasons, and I'm not going to go into great detail about it right now. Ask me at the end. But 2 big things is that extinctions occur at different levels, okay? And we can't always identify everything in a fossil flora to its nearest living relative as a species. Like, it's easier to determine something's genus or even family or order. Okay, So the question here is, what happens when we don't use species? Ideally, we use species, but what if we use genus as the source of occurrence data for these models, okay? Or even something bigger, family, order. What is the effect? And I'm going to go ahead and tell you there's virtually no information on this out there in the literature. So this was kind of a cool study, I thought. All right. So let me talk to you now. I talked to you just now in terms of ecological niche modeling. I did that because we already talked about it. You guys are on board with that. You're comfortable with it. I didn't use ecological niche modeling here to test this hypothesis or to investigate this question, but I use something very similar. So I use something called calibration. Now, calibration works. We're going to start the same way. I'm going to take all the species of extinct species in a fossil flora. I'm going to map them on to their nearest living relatives, find occurrences of the nearest living relatives, extract the environmental data, and then here is where things get a little different. I'm going to find the average of some environmental variable, let's say of mean annual temperature or of isothermality or whatever. And then I'm going to take a flora. Here's a fossil flora, right? Includes only aeschylus and an elm, no equisetum. And I'm going to take the average of these environmental variables of interest and weight them by abundance. So here, 4 aeschylus, 1 elm, to predict some unknown climate. Okay, so that's how that would work. Now, I can also do that so that I work with presence-absence data and the weight becomes 1. And that's how I worked here. All right, so, but how are we going to test this? I mean, you can't really test anything using fossil floors. You could apply it to fossil floors. So in fact, I'm going to stick with living species. And I'm going to find, I'm going to use something here, like rather than an unknown flora, I'm going to use some floras with known climate. So I can determine if my calibration analyses were accurate. So let me walk you through this. Well, where am I going to get some floras with known climates? Well, in fact, I use taxonomic lists from US counties. So 341 approximately counties in the US, give or take a few that have dropped off the map here and there. But anyway, so 341 counties in the US, I obtained taxonomic information for those counties, and that is for living species from the USDA plants database. And for each county, I can know something about its environment, and I obtain that from this World Clim, which represents global climatic data. Now, I divided the US counties into training data and testing data. And so the training data represented 1,521 counties. And I use that to find the average, I use that to find the average of the environmental variables of interest. In fact, 19 environmental variables of interest coming from World Clim here. Then testing data represented 520 US counties. I actually used those averages for each of the taxa to generate some predictions. And so for each of these 1520 counties, then I have some predictions about mean annual temperature, about mean annual rainfall, about temperature during the coldest quarter, these sorts of environmental variables. And then I can compare to what we know to be true, or at least what we assume that World Clim is telling us truthfully about the climate of those counties. And I didn't just do this one time, I did it 4 times for 341 or 3,041 counties, because I'm interested not just in species, but I'm interested to compare the results from species to genera, to families, to orders. Okay, so one way to think about the results here is in terms of one way to think about the results is in terms of their their occurrence within a range of biologically meaningful values. So in fact, to think about these in terms of the numbers that are represented by these climatic variables. And what I mean here is that bio 1 is mean annual temperature, and that's measured in degrees C. Now, is it meaningful if I get a result that's plus or minus 2 degrees C? Probably so. Is it meaningful if I get a result that's plus or minus 10 degrees C? Probably not. okay, for mean annual temperature. But I mean, who knows? I obtained environmental these meaningful ranges from the literature. We could argue about that, but the point is here, what I'm showing you in this graph is the percentage of predictions that fall within these biologically meaningful ranges. So let's take mean annual temperature here, and we'll see that the range is plus or minus 2 degrees C. I think that's biologically meaningful. Again, we could debate that. And we see that 45% of my predictions using orders, calibrations using orders, fall within that range of negative or plus or minus 2 degrees C. If we move across here, we see that families are much improved. 80% of the predictions, or 80% of, I made predictions for 80% of counties within plus or minus 2 degrees C of the actual value. I've highlighted cases where I've correctly predicted 95% or more of counties within these biologically meaningful ranges. And you'll see that genera here outperformed even species. And in fact, if you kind of move your eyes down through these two rows here on the end, genera and species, you'll see in that many cases, almost all, genera outperform species. And that was a highly unexpected result, at least for me. So another way to think about the outcomes here is in terms of the correlation of predicted versus observed values. And that can be measured using R². So in these graphs, which are a little bit hard to see, each of these represents one of the 19 climatic variables. R² is shown here on the y-axis. And in each graph on the x-axis, I'm showing you in order. Okay, anyway, no pun intended there. Order, family, genus, and species. So what I originally would have predicted before I did this study was that you would see here an incrementally increasing line on these line graphs. You would in fact see that orders performed worse than families, families performed worse than genera, and genera performed worse than species. It's not what I see here. I see that in almost all cases, again, like the results I showed you on the previous slide, genera outperform species in terms of in terms of correlation between the observed and predicted values. So what can be said about that? Well, I would say that climatic calibrations performed with genera, and that's the level we often work in the fossil record, okay? Were generally as good or better than those performed with species. I was surprised. Climatic calibrations performed with families were only slightly less good. And climatic calibrations performed with orders were generally inaccurate and imprecise. But here's the thing. I would almost guarantee you that no one is going to go out, collect data from a fossil flora, and perform a calibration analysis such as this one at the level of order. In fact, there's going to be a mix of species, genera, families, and maybe even orders representing the nearest living relatives of the fossils in those fossil floras. So a future direction for this research, maybe not for me, for someone in this room perhaps, who knows, might be to look at what we get when we mix this up. So we mix up the taxonomic ranks. Okay. I'm going to move on and talk to you a little bit about what paleoclimates did Aeschylus experience during the Eocene. Pretty excited to talk to you about this. You've probably heard it from me before, but I'll be working a little bit more on Aeschylus when I start at the Smithsonian. So this question here about paleoclimates experienced by Aeschylus during the Eocene has resulted in one publication with Mona Papish, a collaborator in China, Yundong Gao, and Linda Watson. Where's Linda? Oh, hi. Okay. So briefly, let me introduce Aeschylus to you. In the modern world, Aeschylus exists in mostly temperate habitats, into subtropical regions, slightly in China, but where the distinction between subtropical and temperate is a little bit fuzzy anyway. So trees and woody shrubs that evolved by at least the Paleocene. I'm showing you a time-calibrated tree here, showing you that this root node of Aeschylus, or the Aeschylus evolved by at least the Paleocene here. And since it's been on the scene, Aeschylus is represented in the fossil record by quite a lot of plant parts that span the spectrum from, again, Paleocene into the Holocene era. Fruits, pollen, wood, leaves, you name it. So very good evidence of Aeschylus. Now, when I talk to you about the Eocene, I'm talking to you about this big time period here from 56 million years ago to approximately 33.9 million years ago. And why do we care about the Eocene? Well, so I just told you that in the modern world, Aeschylus is mostly temperate, occurs in mostly temperate areas. Well, the Eocene was a period of death throes from the Cretaceous, where there was a hothouse in the face, like the Earth experienced greenhouse climates or hothouse climates, and so temperatures were really quite far north in the northern hemisphere and south in the southern hemisphere were quite tropical or subtropical in character. And Aeschylus at that time, we know from the fossil record, was occurring with things like bananas and cycads, the things that we typically associate now with tropical climates. So the question is here: did Aeschylus, was it temperate during the Eocene, or is that a new characteristic of this taxon? Okay, so already covered some of this with you. Going to use this methodology, but here I'm using ecological niche modeling, not that calibration. So taking all of the fossils that occur in floras, Eocene floras with Aeschylus, mapping them to their nearest living relatives, getting those occurrence points, extracting environmental data, applying a modeling algorithm. In this case, I use GARP, which is slightly different from that Maxent I showed you earlier. We can talk about that at the end if you wish. Get predicted distributions, stack the models, and extract from that some environmental information. And so additionally, in addition to extracting, well, we'll get there. All right. So first of all, I need some fossil floras from the Eocene that contain Aeschylus. There is no shortage of those, although you guys might not think 4 is a lot. I'm like, yeah, 4. Okay, so there are 4 fossil floras in the Eocene that contain representatives of Aeschylus. They are in British Columbia. 3 of them are in British Columbia. 1 is in Washington State, and 1 is in the island of Spitsbergen, which is an island in the Arctic Circle north of Norway. There's a 5th questionable flora. We can talk about that at the end. I did work with it, but I'm going to gloss over that for the purpose of this presentation. So these fossil floras of Aeschylus vary in their diversity, or their diversity of nearest living relatives, I guess you could say. We worked mostly at the level of genus here. There were a couple of families and cases where we had to work at the level of family and order. So Spitsbergen having 11, I guess you could say, operational taxonomic units that we used, whereas this more diverse flora in Republic, Washington had 63 OTUs. So again, I've already been over this methodology with you. Well, the last thing I wanted to say about that was that in addition to pulling out this environmental information, I shouldn't be pointing because I have remote people. In addition to pulling out this environmental information, we projected these models onto the face of the world to see where suitable habitats for the flora would occur in the modern world, to see if they were temperate, tropical, or so forth. So quickly displaying the results here to you, you will see, showing you at the top, a map of the modern world. Areas in orange and blue represent temperate areas, in green represent subtropical. And you will see that by and large, these data points fall within temperate areas, or areas that would be suitable for these floras in the modern world occur in temperate places. So just mapping these variables onto, or considering the variables that probably have the most influence on where species live, that is mean annual temperature, temperature during the coldest month, and temperature during the coldest quarter. If we think about just, and showing you here the environmental value on the y-axis, and then on the x-axis the 4 fossil floras, and here showing you with these dashed lines a range that we would generally consider temperate for these values. You will see that all of these floras, with the exception of Spitsbergen, tend to fall within the temperate range, maybe a little bit shifted towards the colder end of things. Okay. So, conclusion: Aeschylus most likely experienced temperate or slightly cooler climates during the Eocene epoch. And finally, I want to cover, was there a latitudinal gradient of genus richness of vascular plants during the Eocene and Oligocene epochs? And so that is a study co-authored with Cassandra Walker and my advisor, Mike Palmer. So the latitudinal gradient of richness simply tells us, in the modern world, this simply tells us that this is one of the primary ways in which species are organized, or the diversity of species is organized on Earth. And in the modern world, there's a very high diversity of species towards the equator, and a much lower diversity of species toward, or much lower species richness towards the poles. And that's shown with a stylized diagram here. More realistic looking data here for vascular plants, where I'm showing you in warm colors, high species richness, and in cooler colors, low species richness. Now I'm showing you for vascular plants data for species, but in fact, this is true across higher taxonomic ranks for genera, for families, orders, and so on. So while we know quite a lot about the latitudinal gradient of richness in the modern world, in fact, that topic was something first discussed in the literature in 1807, we know a lot less about it, particularly in terrestrial environments for times in the past. And so this research was concerned with looking at the latitudinal gradient of genus richness, so working at the level of genus because it's convenient for fossils, of vascular plants during the Eocene and Oligocene epochs. Again, Oligocene or Eocene, I'm talking to you about 56 million years ago to 33.9 million years ago. Oligocene, I'm talking 33.9 million years ago to about 23 million years ago. So quickly running through some methodology here, I use macrofossil floras in North America, north of Mexico. And I found those fossil floras, the fossil floras that we aggregated for this study, I pulled from literature sources that had aggregated fossil floras for other reasons. For each fossil flora, I found its richness, and this was a place where biodiversity informatics failed me. I was literally like 1, 2, 3, 4, 5. And also the paleo latitude of each macrofossil flora. So here, we identified 172 Eocene floras with an average number of genera of 15. 29 Oligocene floras with an average number of genera of 22. And to find the paleo latitude, I used an online calculator, which is a new thing that you all can go and try out as soon as you leave here today. I performed linear and polynomial regressions for each time slice, and by time slice, I'm talking about Oligocene and Eocene, to look at the nature of the relationship between genus richness and paleo latitude. And then I compared to modern floras using all the floras from that floras database that I was telling you about earlier. So here's a map made by Cassandra and I, showing you the distribution of the floras that we studied. Didn't mean for that. So 172 of those in the Eocene shown here, and 29 of those in the Oligocene shown here. Although there's a lot less data for the Oligocene, you will see that the coverage of floras in terms of latitude is quite similar. So for modern floras, what we found is that a quadratic or polynomial relationship more precisely or more accurately described the distribution of genus richness within the study area, so North America, north of Mexico here, with a modeled peak in genus richness at about 31.5 degrees north. That's like the panhandle of Florida, I think. Also, looking here, you will see that here on the bottom of the x-axis, we have midpoint latitude. On the y-axis, log number of genera. This bottom graph is showing you log genera on the y-axis again, and log area. So area being a very well-known predictor of richness, of patterns in richness, log area on the x-axis, and a much stronger correlation between area and richness than latitude and genus richness. And that's important because we can't know area for fossil floras. So here, results from fossil floras, we actually find, again, like with the modern floras, the quadratic relationship or the quadratic model best explains the genus richness along lines of latitude. So log genera here and latitude here at the bottom of on the x-axis, and showing you peaks in diversity or modeled peaks in genus richness at 45 degrees north for the Eocene and at 48 degrees north for the Oligocene. So you will see in both cases, these peaks in diversity were shifted northward. And I will take one moment to hypothesize. I'm still kind of processing this data, but I'll take one moment to hypothesize and say that during the Eocene, that's because temperate or tropical climates reached much further north than in the modern time. In the Oligocene, that represents a period of temperate diversification. And so I think both of those could explain, assuming that these models are accurate, those could explain why we're viewing differences or why we're seeing differences here between these time slices and the modern floras. So Main conclusions: flora in the Oligocene and Oligocene had hump-shaped latitudinal gradients of genus richness. These gradients may be steeper or stronger than in the present day. The peaks of genus richness in North America and north of Mexico, maybe further north in the Oligocene than in the modern day, and even further north in the Oligocene. And among modern flora's area, as we would expect, shows a stronger correlation with genus richness than latitude, and that's probably really important for explaining past diversity, although we can't know area for fossil floras. Okay, so here we are at the end, summary of the whole talk in 4 bullet points. Don't use ecological niche models to predict species richness, or be wary of doing it. Use more genera and species, fewer families, and even fewer orders when doing paleoclimatic estimation using calibrations. Aeschylus was temperate during the Eocene, and the latitudinal richness gradient of North America and north of Mexico was likely hump-shaped during the Eocene and Oligocene time periods. All right, so here at the very end, I gave some acknowledgements throughout this presentation. However, this is a highly appropriate place to acknowledge the entire botany department for supporting me throughout these 5 years and for making my life amazing. My lab, current members and past, Libra, and finally, the High Performance Computing Center, in which I conducted many of these analyses, except those ones where I told you I had to count 1, 2, 3, 4, 5. These sources of funding who have made my graduate career, or that have made my graduate career really awesome. And finally, I want to leave you with this to tell you that where I'm going in the future. So right now, I'm finishing up doing some analyses of weedy Brassicaceae and Solanaceae for a weed flora of Oklahoma. I've been sitting on some data that is an update to the phylogeny of Aeschylus that I need to publish. So that's represented up here by a phylogenetic tree. And lastly, down here, I am showing you this rather complicated figure of the work that I plan to do at the Smithsonian. I will let you read that for yourself. It pertains to the assembly of the woody flora of North America. So for the remote viewers, I am glad to take any questions that you have through email. And otherwise, I'm going to sign off now and ask the rest of you if you have some questions or comments.
Thank you, AJ, for your talk and for impeccable time. We do have some time for questions, realizing that people do need to leave for classes, and also AJ and her committee needs to leave